Hi everybody, it's good to see you all. I'd like to tell some more today about what DNA technologies can actually assist plant breeders to make these beautiful flowers for the future. When I was a child, I got interested in plants, fascinated by them. I saw them growing in the grass, and I studied their flowers. They were small and pink and with 5 petals, and sometimes they were red with 4 petals or yellow, and all the colors and all the different shapes. Sometimes they smelled even beautifully. And I wanted to know what their names were. They had names. So I looked it up in my booklet. And I got to know them one by one, and sometimes I got confused. You also have flowers that really look very similar. And then you have to look at the small differences. And that got me going. I still do this. So it gets me going. I really, really like that. And then I also saw that flowers in the shops, they're similar to the ones that you can find in the field. How's that related? I wanted to know more about flowers, basically, when I was younger. So I decided to study biology. I thought that was a good choice, to learn more about how they grow and how they develop, why they are different, what's going on there. In my study, biology, I also got, practicals. You got in the laboratory. And then you start to look at more details. You start to look at the DNA. You cannot DNA with the eye. You need to do special technologies to make it visible. Basically, that's all what you do. You do things in the lab and make DNA visible, and then you can, base your conclusions on that. Another thing I learned, that was genetics. You also have genes. Genes determine what kind of phenotype a plant will get. And of course, the genes are encoded by the DNA. So there came this whole spectrum of things I really liked. It was plants, DNA, and genetics. So it's a good job then to become a molecular plant breeder, right? So that's why I went on in studying DNA technology. And with this technology, it is so nice if you can look at the DNA and you know the traits. You can help plant breeders to breed faster, to get better to the traits they are interested in. And one of the challenges that plant breeders have in breeding the new varieties is that they need to be resistant or tolerant against all kinds of pests and diseases. Most of them are caused by aphids, thrips, there are insects, or by fungi. We see the powdery mildew, and we see rust there, but there's also bacteria, nematodes, and viruses. There are a lot of pathogens that the plant needs to be resistant to, because we don't want them to look like this, right? You can spray with chemicals. We don't want it anymore. It's not sustainable. We need to go for the resistance of the plant itself. And where is it? Can we find it in the DNA? Another thing is also, of course, climate change. It gets drier and hotter, or it gets cooler and wetter. Yeah, now I have to think. So it's a whole range. It gets more extreme. Also, plants need to be adapted, especially the ones that we grow in our greenhouses and in our garden. They're really pampered. They're not the natural plants anymore. They're more beautiful and more valuable for us in the garden. So we need to adapt those plants better to all these kind of situations. And again, I can tell you, if we know more about the genetics of these kind of traits, it's easier for the breeder to build it in. Another challenge, what I like next to the plant itself, is of course the genetics that how's the genome composed? How does it look like? In this case, I want to show you what genome size is. Humans have about a genome big as 3.2 gigabases, 3.2 billion base pairs. Now, the maize plant that you see on the picture has a genome which is about the same size as a human. It is 2.3 billion base pairs. The tulip, on the other hand, is a lot smaller, but it has a genome that's 10 times bigger. Now imagine if you want to do genetics with that. It gets really, really complicated, the breeding and crossing. Because another thing that it is, if you have a large genome, also your life cycle gets longer. For a tulip, if you go from one seed to the next generation, so again, seed, it takes 6 years. So if you want to breed a new variety with some special traits in it, and you have to make 5 crosses and select the offspring, it takes you 30 years. So imagine, you really have to think into the future as a breeder, because you have to think now of the tulip that you will need in 30 years. It's almost undoable. Another challenging aspect of plants is that they are polyploid. Not all of them, but some can be polyploid. And we have 2 copies of our genome, one from our father, one from our mother. And then we are diploid, but plants can have up to 4 copies. Here you see a plant with 4 copies. You can have 6 copies, and then we have a chrysanthemum. You can have 8 copies, and then you're a strawberry. Having more DNA or more genomes means, again, you get bigger. And you have a more complex genetics. Instead of 2 copies of 2 alleles of a gene to segregate, you have 4, one parent, and 4 different one in the other. You do the math. It's complicated. But it's also nice. It's nice to work and find ways that we can simplify it. And that can be done by looking at the DNA itself, look at the genes itself. If you know which copies of these 8 possible that you have in the population is the one that gives resistance, you can make a DNA marker for it, and you can just select it. Very easy, by very easy and simple PCR test. So what we can do with our work in a laboratory is we can look at the DNA, we can follow the genetics. What else we can do is we can study the parents, see what kind of traits they have. So you've got to have a good idea of your parental population, how they look genetically, and then basically you can design your own plant. You can think, oh, I need that trait from this parent, that from that, and that from the other one. Make ingenious crosses and select easily on the DNA level if the traits are present in which of all the offspring plants you have. And then you can take that one out and use that for further crosses. So it's basically designing your own plant through the DNA. Now, what technologies are we using for this? The first one that I will mention is molecular markers. With molecular markers, you can look at a small piece of the whole DNA molecule. You don't need to know all the DNA, only the ones that code for certain traits you're interested in. So molecular markers are a very easy technology to look at a limited number of traits you're interested in and see if these markers all come together in that one offspring plant, and you can use that for the further crossing. We also call it DNA fingerprinting. Now this technology, next to that it's being used a lot these days, especially in vegetable breeding, not that much in ornamental breeding yet, but things will come, is that the technology has improved tremendously over the last 20 years. When I started this, when I was a student, with the molecular markers, it took me a whole week to look at one marker. It was a very old DNA technology. I will not mention it. But it took me a whole week. Nowadays, I see students who go in the lab and they do 100 traits on 100 plants, basically in an afternoon. So that's a tremendous improvement, technology. You can see that technology improves it. It gets more easier, it gets faster, and it gets cheaper also. If you can do things faster and easier, most of the time it's also getting cheaper. And you can do a lot of samples at the same time. High throughput screening. Now take these bikes, just to give you an idea what technology improvement means. What's the difference? Can you see that? I'm not talking about the color of the saddle or something, or the wheels color. What's the difference between these two bikes? You probably have the left one still. My parents have the one on the right. It's an electric one. They cannot cycle anymore on the old one. They're too old for that. But with the other ones, they go. And that's nice about this technological improvement. You can do more things than you did before. It's faster, it's cheaper for your energy going on an e-bike. It's easier, and you can do more mileage. High throughput, you can also call that. So my parents can still cycle now. Otherwise they couldn't. And it keeps them healthy and fit, so it has a lot of beneficial advantages. Another technology that has seen a tremendous innovation is DNA sequencing. With DNA sequencing, you determine the letters of the DNA. And after you did, well, you get this information that doesn't mean anything yet. You need someone who's schooled in bioinformatics to help you and find the genes, find the information, when the genes are expressed, how they are expressed, and where and how long, and get all that specific information also there. And it's a lot of information that you get from one genome. The human genome, I told you, 3.2 billion base pairs. If you would write it down in a book, you would need a whole bookcase to fill with all the letters of the human genome. So that's a lot of information, and not everything goes for genes, but that's another story. And now think of the tulip. It gets you 10 of these bookcases, so that's a lot of information. But if you only look for that one trait, you could say, I'm looking for a needle in a haystack, which it is, but now we have a magnet to pull the needle out, and you can see if it's there. And the magnet is a DNA technology. Marker technology. And if you want to do more than one trait, it becomes more, efficient and cheaper to do DNA sequencing instead of running 10, 20, or even 100 markers. If you go to 100 markers, well, then I would say, why not sequence the whole genome? That's fast, easy, and cheap as well. About the money, the first human genome that got sequenced 20 years ago, and that cost $3.2 billion. So $1 for each base pair, basically. It took 15 years. Now I come to the next one. Now we have a technology. It took those machines on the left-hand corner, big blocks, machines, first automatic sequencers, and 300 of them working day and night. And after 3 years, basically, we had all the DNA sequences we needed to construct the whole human genome. And now I have this really small, you can see it in the lower corner, a really small apparatus, just the size of a microscope glass. And you just hook it up on your laptop, and it does the whole human sequence for under $1,000, and just in an afternoon. So that's a tremendous increase in speed and efficiency and technological improvement. And I think that's also very fascinating in this work, that you have also all these new technologies coming up all the time and see, and then also if it works better and how it's improving things, and you also have to fine-tune things before you can get it really to work in your crops. And I think that's the fun of it. So on the right-hand side, I'll give you a glimpse on how it works, this nanopore technology. Through a very small hole, a strand of DNA is pulled, and the hole is in a membrane, and the membrane has an electrical current on it. And through that electrical current, the disturbance of this current, by the different nucleotides, you can tell which nucleotide came through. And this goes very, very fast. You cannot even tell how fast it goes. I mean, in seconds, it's working. And what's more, now you see one hole, but in reality, it has thousands of holes in the same membrane. So simultaneously, you can sequence thousands of DNA molecules. So this is a nanopore technology. And I also have one in my laboratory. And it's very nice to do all these new sequencing projects with it. But there's also new technologies that come up. These two ladies, I see some of you nodding. They are Jennifer Doudna and Emmanuelle Charpentier. They received the Nobel Prize just 2 years ago for their technology, for the invention or the discovery, I better say, of CRISPR-Cas technology. And this is something we might be able to use in the future to aid plant breeders. With this technology, we can make mutations at exactly the spot in the genome that we like to have a mutation in. And a mutation is not a bad thing. Mutation is good because it creates genetic variation. And genetic variation is the basis of evolution, but it's also the basis of plant breeding. If you do not have genetic variation, then there's nothing to breed with. So with this technology, we can pinpoint the place where we want to have a mutation, and it's compared to older technology. Again, this is a technological revolution. Also, with older technology, we could also make mutations, but then you would make a whole range of mutations in the DNA, and we call that random mutagenesis. And then you had to hope for, well, the event that the mutation was in the spot you needed, and now we can actually put it directly in a good spot. And it's still in development, this technology, and it's not used in plant breeding so far, but maybe in the future we can, and I think that would be awesome. So I've showed you the fascination of plants, how we can make them more beautiful and more sustainable in the future, and how DNA technology is also very fascinating, and how it can help and assist the plant breeders. So who wants to join me? Solve the genetic puzzle and make the new sustainable plants of the future. Thank you.